Today we will discuss about the proteins. What are proteins and classification of proteins and structure of proteins. As we know that the amino acids are the building blocks of protein or basic unit of protein. Proteins are important for our proper growth and maintenance of the body, and there are different parts of our body which are made up of proteins, just like our hair, nails and teeth and etc. In nature, as we know that there are different types of amino acids are present, more than 700 are in nature. Out of them only 20 are present in the formation of, which participate in the formation of proteins. And we have already discussed about these 20 types of amino acids. And the proteins, how form the proteins? Proteins can be formed by the joint or condensation of two amino acid molecules which are linked together by the help of peptide bond, which are formed between carboxylic acid group and amino group of two amino acid molecules. For example, as we know that this is the structure of one amino acid molecule. Here is presence of amino group, carboxylic acid group, one carbon which is also known alpha carbon, one hydrogen atom, and this one is side chain or variable group. Variable group means here can be one hydrogen atom, one methyl group, one aromatic ring. Any type of group can be attached at this place. Now, these two amino acids, when they join together or when the condensation becomes in between of these two molecules of amino acid, then one water molecule come out and the peptide linkage is formed, and the bond which is formed between carboxylic acid group and amino group, between the oxygen of carboxylic group and nitrogen of amino group, is known as peptide bond. And the product is known as peptide. This linkage is also known as peptide linkage or amide linkage. Same, when more than two amino acids or more than two, that can be 3, 4, 5, the linkage will be formed and it becomes more peptide linkage, just like this, and then forms a linkage of polypeptide. Polypeptide, also known as proteins. Here amino acid molecules can be similar or differ. Here can be aromatic amino acid, basic amino acid, acidic amino acid. Now comes on the structure of proteins. As here we see, there can be similar or different amino acid molecules participate in the formation of protein. So the structure complexity on the basis of these amino acids and complexity of the ring formation and protein formation, structures can be studied into four types of studies. There can be primary structure, secondary structure, tertiary structure and quaternary structure. In primary structure, this structure is very simple and can be helical and can be in plate form. It is a linear structure. This formation of linkage is also, we can say that, a primary structure. This is only linear in formation, and there is no branched, there is only simple peptide linkage which is formed in a linear formation. Next, when we come on the secondary structure, it can be defined by two types of structure. One is alpha helix and second one is beta plated sheets. In alpha helix, there is a helical structure between the amino acid molecules and one hydrogen bonding by which the helix is formed. There is a hydrogen bonding in between two molecules. This one amino acid and this one amino acid, there is a hydrogen bonding. The helical structure arises due to resonance in the peptide linkage and hydrogen bonding between NH and CO, between the hydrogen of amino group and carbon of oxygen of carboxylic acid group, by which the turn of helix are formed. In the alpha helical structure, each turn of helix has nearly 3.6 amino acids and its distance is 5.4 angstrom. Between these amino acids, the distance is 5.4 angstrom, and the amino acid molecule which are present is 3.6 amino acids in one turn, and the diameter of the helix is 10 angstrom. This diameter is 10 angstrom. Now comes on the beta plated sheets. These are this type. These can be straight, these can be horizontal, vertical. These are beta sheets. These are sheet-like structure, hence name as beta plated sheets. This type of structure was first proposed by Pauling and Corey in 1951. Now we discuss about beta plated sheets. They can be parallel or anti-parallel, depend on the amino acid molecule which run in similar direction or opposite direction. In this structure, the polypeptide chains is extended and chains are held together by the intermolecular hydrogen bonding. Two types of plate sheets are present in the protein structure. It can be parallel or anti-parallel, depends on the molecules runs in the same direction, that known as parallel sheets, and if they runs in the opposite direction, they known as anti-parallel sheets. If they runs in the same direction, this is parallel sheet. If they runs in opposite direction, then known as anti-parallel. This is anti-parallel. Now comes on the tertiary structure of proteins. The tertiary structure of protein represents overall folding of the polypeptide chain, that is further folding of the secondary structure, which can be alpha or beta sheets. It gives rise to two major molecular shapes, that is fibrous and globular. The main forces which stabilize the secondary and tertiary structures are hydrogen bonds, disulfide linkage, van der Waals forces and electrostatic forces. Now comes on quaternary structure of proteins. Some of the proteins are composed of two or more polypeptide chains referred to as subunits. The special arrangement of these subunits with respect to each other is also known as quaternary structure of the protein. This structure is more complex than other structures, and its complexity is rather than other structures just like primary, secondary and tertiary of proteins. Now comes on the classification of proteins. On the basis of increasing complexity and solubility, they can be classified into three different classes. One is simple proteins, second is conjugated proteins, and third is derived proteins. Now comes and discuss about the simple proteins. They gives alpha amino acids on hydrolysis. Just like if there is primary structure and linear structure, there are only alpha amino acids are participates in the formation of linear polypeptides, hence known as simple proteins. So they gives alpha amino acid on hydrolysis, for example albumin and globulin. They can be further classified on the basis of decreasing order of solubility. First one is albumin. They are soluble in water, acid and alkali and coagulated by heat and precipitated by the treatment of ammonium sulfate. Now second one is globulin. They are insoluble in water but soluble in acid and alkalis and coagulated by heating and precipitated by reaction with ammonium sulfate. Now third one is prolamines. They are also insoluble in water and salt solution but soluble in dilute acids and alkalis and 70 to 90% ethyl alcohol. For example, Jane, which is present in maize, and gliadin, which is present in wheat, and hardin, which is present in barley. Next one is glutalins. Fourth one is glutalins, which is insoluble in water and dilute acid but soluble in dilute alkalis and coagulated by heat. For example, glutabin, which comes from wheat, and oraisenine, which come from rice. Now fifth one is albuminoids or scleroprotein. These are insoluble in water and salt solution and soluble in concentrated acids and alkalis. For example, creatine, which are present in our hairs, and fibrin, which are present in fibrous proteins. These are also of two types, collagen and elastin, which we know that they are present in the cells. Next one is an important histone proteins. So they are soluble in water and dilute acid but insoluble in dilute ammonia and cannot be coagulated by heat. They are rich in histamine and arginine amino acids. Last one is protamines. As our name indicates, they are soluble in water, dilute acids and alkalis, ammonia. They are more basic than histone proteins. They have a simpler structure and cannot be coagulated by heat. Now comes on the second type of proteins. These are conjugated proteins. They contain simple protein molecule with non-protein groups. On hydrolysis, they give non-proteinaceous groups. These non-proteinaceous groups are also known as prosthetic groups. As we know about enzymes, they are proteins and contain prosthetic groups, which are non-proteinaceous group. So enzymes are also known as proteins, but not all enzymes are proteins. All proteins can be enzymes, but all enzymes cannot be proteins. For example, riboenzyme. These are made up of RNA, ribonucleic acid. Hence, all enzymes are not proteins, but all proteins can be say are enzymes. Now comes on the conjugated proteins. They can be further classified into following classes. First one is glycoproteins. As the name indicates, they contain the carbohydrate groups as the prosthetic group or non-proteinaceous groups in their formation. For example, mucin of saliva. Next one is phosphoprotein. In this protein, the phosphorus is present as a prosthetic group in proteins. And for example, casein protein of milk. Next, third one is lipoprotein. As the name indicates, they contain lipoproteins of serum and they contain phospholipids. These are a part of lipids. And next one is nucleoprotein. As the name indicates, they contain nucleic acid as the prosthetic group in their structure. For example, nuclein in dead pus cells. Next one is chemoproteins. These contain or characterized by the colored pigments. For example, chlorophyll. They contain pigments as a prosthetic group. Now comes on the third one type of proteins. This is derived protein. These are formed by the action of physical, chemical or enzymatic agents and can be further classified on the basis of cleavage of proteins. First one is denatured proteins. As the name indicates, they are the denatured protein and insoluble and formed by the heat of protein, by the action of heat on protein. Second one is primary protease or metaprotein. These are also insoluble in water and dilute solutions and soluble in acids and alkali. Third one is secondary protease. These are soluble in water, not coagulated by heat and precipitated by saturation with ammonium sulfate. Fourth one is peptones, polypeptides or simple peptides. These are soluble in water, neither coagulated by heat nor precipitated by the saturation with ammonium sulfate. On the basis of the molecular structure and behavior, proteins may be classified on different two types of proteins. One is globular protein and second one is fibrous protein. Now we comes on the fibrous protein. They are generally linear condensation product of natural amino acid. As the name indicates, fibrous, there are no branching and cross linkage in their structure, only long linear polypeptide chain and are held together by intermolecular hydrogen bonding. They are insoluble in common solvent and soluble in strong acid and bases. Second one is globular protein. As the name indicates, they are globular or spherical in structure, hence very branched in structure, highly branched cross linkage and condensation product of basic and acidic amino acids. These proteins may also be of 2 or 3 dimensional shapes. These polypeptide chains stabilized by intermolecular hydrogen bonding. These are soluble in water.